Okay, in this section we're going to start off our muscular system examination by talking about the types of muscle tissue. And the first type of muscle tissue we're going to look at is smooth muscle. And smooth muscle is not under our voluntary control. We do control it through the central nervous system, but we actually don't control it voluntarily. They are spindle-shaped cells that have one very large, prominent nucleus. Basically, spindle shape means that it's kind of tapered at the ends, almost kind of pointy, and then a little fatter in the middle. They have no alternating pattern of light and dark bands, and muscle cells that do have this are called, they're called striated because those bands are called striations. Most of them can actually function without direct nerve stimulation. They actually respond to changes in the environment. And examples of locations that have smooth muscle are places like the stomach, the digestive tract, sphincter muscles like within the digestive tract, as well as within our blood vessel walls. Now the second type of muscle that we have is cardiac muscle. And cardiac muscle is found only in the heart, and it functions to pump the blood. These are very small cells. They are striated, so they have that alternating light and dark bands. And when you look at these under the microscope with the lab that we're going to do, the striations are so tiny that you actually really can't quite see them with our microscopes. But these cells only have one nucleus per cell, and you will be able to see those. They do branch like branches of a tree. If you think of, a branch goes up at a tree and it splits, that's what happens to these cells. Where they connect to other cells is a special site called an intercalated disc, and we will be able to see these under the microscope. Again, it's not under the direct control of the central nervous system, and this is actually a good thing because otherwise certain procedures that we do, like heart transplants, would not be possible if we had to reconnect a bunch of different nerves. The other nice thing is it does not fatigue like skeletal muscle, so that is a characteristic of it. Our heart actually pumps from before the time we are born until basically we die, and if it got tired, we'd be in a whole lot of trouble. The third type of muscle, and the one that you're most familiar with, is skeletal muscle. And skeletal muscle is usually attached, of course, to the bones. That's where it gets its name. They are responsible for voluntary movement as well as production of heat. And so when you shiver, when you're cold, what you're actually doing is contracting your skeletal muscles and producing heat to hopefully raise your body temperature. Most of them are controlled by the central nervous system. They also have striations, so you will see, it's actually kind of purple on the slide, an alternating pattern of dark purple, light purple bands with the skeletal muscle sample. And we also have many nuclei per cell. Now, unlike the cardiac muscle, these cells aren't branched, and you will see this with the fibers. They actually are in strips that kind of run horizontally. They don't split off and join and mesh with each other. Examples of skeletal muscles include the bicep, the tricep, and the quadricep. Now, in terms of the structure of our skeletal muscle, most of our skeletal muscles attach at two bones, and they go across a joint because that's what actually permits movement. Now, one of the bones is usually more stationary for a movement than the other. Like if you flex your arm, my bicep muscle is actually attached to both my upper end of my humerus and my scapula, as well as my ulna on my lower arm. Now, obviously, my humerus, when I flex my arm, doesn't move a whole lot, but my ulna, my lower arm, does. The stationary end is called the origin, whereas where the muscle attaches at the movable end is referred to as the insertion. Now, how it's connected to the bones is with a tendon. And in addition, we also may have something called the bursae, which we talked about with the joints, that help prevent friction. Again, that is joint dependent. Now, in terms of the structure of skeletal muscle fibers, the muscle fiber is actually a contractile cell of muscle tissue. And we'll actually do a graphic organizer here in a little while that will show this. The basic unit of structure is something called a sarcomere. So we put sarcomeres together to create a muscle fiber. Okay. Now, within the sarcomere, we have thread-like structures that make up a sarcomere. We have the thin filaments that are made of a protein called actin, and we have the thick filaments, which are made of a protein called myosin. And what happens is those thin and the thick filaments actually interact with each other to produce a contraction. Now, sarcomeres themselves have a structure, and they are separated from each other by a structure called Z-lines. Now, basically what happens is these sarcomeres are kind of attached, almost like stitched together end to end. And you will see that on the diagram that we do here in a second. The thick filaments are held together at a place called the end line. And the arrangement of these sarcomeres is actually what creates that alternating pattern of light and dark bands, the striations. If we line up the sarcomeres, not only stitch them end to end, but stack them so that all of these areas are lined up on top of each other, then we get this banding pattern. The A band is the area that contains the thick filaments, and the way I remember it is the word dark, because dark has an A in it. Whereas the light area is called the I band, and that is the area that has the thin filaments, because obviously thin, you know, if you write a thin line with your pencil versus a thick line with your pencil, which one is going to be darker? And I remember that with the word light. There is also one other area, it's called the H band, and that's the area where only the thick filaments exist, and there's no overlap there with the thin. And I'll show you that here on our first diagram of the sarcomere. All right, let me just get my writing utensil. Okay, so this is a sarcomere, and a sarcomere runs from this structure all the way down to this one, and you can see how they are actually kind of stacked one after the other and attached at this area. This is the Z line, and so this is also a Z line. So one sarcomere runs from Z line to Z line. Okay, and it just repeats. Now, if I want to have striations, I also have to stack them above and below this sarcomere to get the areas of light and dark to match up. Smooth muscle also has sarcomeres, and they are stitched end to end like this, but because they're not stacked up one on top of the other, we don't get that banding pattern. Okay, this area here with the purple is our A band, because as you can see, the purple area is very thick. Those are our thick filaments. So if those are our thick filaments, then this must be our M line, and you can see the M line is literally in the middle. Now, if that's the A band, then these red here, okay, are actually our thin filaments. So we get this area that actually runs between two sarcomeres here, and then we'd have another one over here, okay? These are our I bands. So we have one I band here and one I band here. So if I'm looking through a microscope image, I'm not going to be able to see the Z lines unless I use an electron microscope image. But basically, the sarcomere runs from the middle of one Z line, or middle of one I band to the middle of the next I band. The only thing that that leaves us with is the H band, and the H band is actually already marked here. This is the H band. And you can see it's got area where we've got none of those thin filaments here. It's just the thicks. So that's why it's called the H band. Now moving on to the next one. This is an electron microscope image of another muscle fiber, and we're going to label those same things here. Now this one, because it's an electron microscope image, we can actually see both Z lines. And in the middle, we can see the M line. Now when we look, we're not going to be able to see either of those things. So once again, one sarcomere runs from Z line to Z line. Now you can see here we definitely get this light area. Okay, that's an I band. We have another one down here. Then we have our dark area, which is the A band. And you can see that we've got this darker area here within the A band. Then we have this light. It's still dark, but it's lighter than on the sides. That's our H band, where we only have the thick filaments, not the thin. All right, the thin filaments themselves, let's talk about those for a second. They are made up of twisted strands of actin molecules, and one side of these actin molecules has what we call an active site, and those interact with the myosin on the thick filament during a contraction. Now, when the filament is at rest, the active site is covered by strands of another protein called tropomyosin. And it's held in place by troponin molecules that almost act like little clips that hold those tropomyosins in place. When the active site is exposed, the myosin and the actin will automatically kind of get drawn to each other magnetically and stick together. Now, when that happens, we're going to get contraction, and obviously we don't want contraction all the time. So we have to have a way of preventing that. And taking a look at our diagram, okay, those red twisteds are the thin filaments, and you can see we've got them, and we're going to take a close-up here of them. And those round-shaped blobs, I guess so to speak, are the actin molecules, and the yellow area is the actual active site where they would interact with the myosin. Now the blue strand is the tropomyosin, which covers up those active sites, and you can see the brown troponin kind of hold them in place over the top of those active sites. The thick filament is made up of myosin bundles, and these always remind me of flowers, like a bouquet of flowers in terms of how they are arranged. They have a tail as well as a globular head that pivots on a hinge, and it's kind of like how your wrist pivots back and forth. That's how it pivots. Now they are bundled, like I said, like flowers in a bouquet, so that their tails are all grouped together, and the heads point up or down towards the thin filaments. So the heads point away from the end line, kind of held together by their tails in the middle, and then the heads actually point out towards the thin filaments. So you can kind of see that arrangement here, where all the heads are pointing away from that end line. The end line is kind of the chain-looking thing that's holding them together. And then the heads are kind of sticking out almost like buds or flowers. Okay, if we take a look at a single myosin molecule, you can see it's got that nice straight tail, and you've got that hinge area that allows that pivoting action that they're talking about with the thick filament. Okay, that is it for our types of muscle tissue. What we will do next in the next segment is actually talk about the contraction process. And in the meantime, I suggest you check out on Moodle the flow chart for the concept map, or the graphic organizer that I sent to you about skeletal muscle structure. It just gives you kind of an idea in terms of the organizational levels for them.